Hi, and welcome back. In this section, now that we've learned the structure of all the tubules in the kidney, we're going to talk about a little bit of function. So let's start with the filtrate. Once it has exited Bowman space, it enters the proximal convoluted tubule. And this is the part of the kidney where reabsorption of the vast majority of the contents is going to occur. So we have this massive amount of fluid filtrate, and we are going to send lots of the water back. In fact, more than 2/3 of it through the process of osmosis. We're also going to be sending back amino acids, bicarbonate ion, the vast majority of the sodium. And we will send back glucose as well, thereby leaving us a much smaller pool of solutes to travel in what's left of that filtrate. Now, the glucose transportation is a little bit different than the other ones in that glucose molecules require a transport protein. And so if you have normal levels of glucose and normal levels of transport protein, it's hunky-dory, all the glucose goes back. But if you have elevated levels of glucose as seen in diabetes or other conditions, you don't have elevated levels of transport protein. So only that amount of glucose for which you have transport proteins will go back into the bloodstream, and the remaining part of the glucose stays in the filtrate where it will have an osmotic pull to keep some of the water in the filtrate rather than being reabsorbed. And this will also change the specific gravity of the urine, as well as give you that positive test for glycosuria and makes the urine sweet. After the proximal convoluted tubule, what's left of the filtrate will move into the loop of Henle, where generally water and sodium are returned to the blood. There's a lot more detail involved in this in a later discussion. Now, when you look at the loop of Henle, what you will notice on both the ascending and descending limbs of the loop of Henle, there is a thick segment. And the reason why it's a thick segment has to do with the epithelium. Here we have a simple cuboidal epithelium. And this epithelium has lots of mitochondria, so it can make lots of ATP because we are going to have active transport of salts occur in this area. In contrast, closer to the loop end of it, the U-turn end of it, these are the thin segments of the ascending and descending limbs of the loop of Henle. And they're called thin segments because the epithelium is thinner. In other words, it's a simple squamous epithelium. And this has a much lower level of metabolism. So we're not going to be having active transport occurring here, but it is very active because of some structures making it very permeable to water. After the loop of Henle, we move on to the distal convoluted tubule. And here we're not really dealing with reabsorption anymore, but this is kind of like a second chance place for secretion to occur from larger molecules which could not get through the fenestrated capillaries of the glomerulus. They now have a chance to enter the filtrate in the distal convoluted tubule. So these are going to be cuboidal cells, just like we had in the proximal convoluted tubule, but they're not going to have the microvilli for increasing surface area, and although they do have mitochondria, they don't have the same number of mitochondria as seen in the proximal convoluted tubules. Now, in the collecting duct, we have two very disparate types of cells. And so the most abundant, in other words, the principal cell, is called the principal cell, where it is spelled the same way as my last name, P-A-L at the end, not P-L-E. And so this is the more abundant cell type, and it has hormone receptors. So first, let me talk about a hormone we've heard about before, and that's parathyroid hormone. So yes, there's parathyroid hormone receptors here in the collecting duct, but they also are parathyroid hormone receptors in the convoluted tubules and loop of Henle as well. So when parathyroid hormone binds to these renal tubular cells, that is going to cause a reabsorption of calcium from the filtrate, thereby increase blood calcium levels. All right, but that's not the main reason why I want to talk about hormone receptors in the principal cells. It has to do with they have receptors for aldosterone and antidiuretic hormone, which we've already learned about, as well as our two natriuretic peptides, which we'll learn about shortly. And so as we know, aldosterone and antidiuretic hormone, they all cause sodium and water reabsorption to increase blood volume. And natriuretic peptides will affect the sodium and water concentrations. They just are not going to act in the same manner as aldosterone and antidiuretic hormone. So stay tuned to learn more about your natriuretic peptides. Now, besides the principal cells, we have a smaller amount of a second cell type. So when we look at the principal cell, we can see that it looks cuboidal, and in the larger collecting ducts, it actually could be columnar in shape. So the other cell type is always going to be shorter. They're not really a squamous cell, but they're kind of not a cuboidal cell either. They're kind of in between. And then they also have surface projections, which the principal cells will never have. And these cells are called intercalated cells. And the intercalated cells, their job has to do with acid-base balance. They are going to be secreting hydrogen ions. So to get rid of extra acids. So here's what happens in chronic kidney disease. Those intercalated cells, which are responsible for acid-base balance, will transition to a cell type called transitional cells because of the signaling changes that happen in chronic renal disease. And then after their transitional cells, they will actually transition and become principal cells, meaning you will be losing your acid-base balance. So if you look at the second row on the photomicrograph, you can see some intercalated cells with their surface projections, and then you can also see some flat cuboidal cells, which are principal cells. Now, yes, I did take a picture that showed more of the intercalated cells so you could specifically see them, but in reality, there's at least 2 or 3 principal cells for every intercalated cell. And so in the cartoon drawing, you can see some of those intercalated cells becoming transitional cells. And as the renal disease progresses, what you end up is with a collecting duct that is all principal cells. So at this point, you can no longer secrete any hydrogen ions to maintain acid-base balance. And so patients with chronic kidney disease have a chronic acidosis as a result of this. So take a moment here and review all the different types of epithelial cells we have learned about, starting with the glomerular capsule, which is a simple squamous, as well as glomerulus itself. But in the glomerulus itself, we have those podocytes. Then we move on to the proximal convoluted tubule with its cuboidal cells with abundant microvilli and lots and lots and lots and lots of mitochondria, so lots of ATP is there. Moving on to the loop of Henle, we have cuboidal cells in the thick segment and squamous cells in the thin segment. In the distal convoluted tubule, we are once again back to cuboidal cells with mitochondria, although less than we had in the proximal convoluted tubule. And most noticeably, there is an absence of microvilli at this point. And then we move on to the collecting duct with both its intercalated cells and its more numerous principal cells. So putting all these functions into kind of like a Sparks Note review, the glomerulus is where filtration is going to happen. The proximal convoluted tubule is going to be doing lots of reabsorption of water, ions, and other solutes, including sodium and glucose. Then the loop of Henle has to do with salt and water being sent back out of the tubules. The distal convoluted tubule has the new function of secretion, and then the collecting duct has both water and sodium changes occurring, as well as pH maintenance. So take a minute, stop the video, look at these last 2 review slides, and I will see you in our next class. And have a great day. Thanks.